Hello, wonderful people. What is going on? It's Medicosis Perfectionalis, where medicine makes perfect sense. Welcome to the first video in my biochemistry playlist, where together we're going to study the chemistry of life. What is your diet? Your diet has fat, protein, and carbohydrates. Your body uses metabolism to deal with these foods. You can build stuff up called anabolism, or you can break it down called catabolism, utilizing lots and lots of enzymes. Enzymes are catalysts. They increase the speed or the rate of the chemical reaction without being consumed by the reaction. Click the like button, click the subscribe button, and let's get started. This is my biochemistry playlist. Please watch these videos in order for maximum understanding and retention. I have a video for every biochemistry topic that you can imagine. By the way, I have two biochemistry playlists on this channel. One short and one long. If you're studying for the MCAT or the DAT or the NEET exam, etc., watch the short one. But if you want to be a doctor, a nurse, a professional, watch the long one. The short one is titled Biochemistry for the MCAT. The long one is titled Biochemistry New, as in Neogenesis. Your diet has carbs, has proteins, has fat. When you eat big molecules. And then through digestion, you'll break down the big parts into smaller parts. So the carbohydrates, whether starch or glycogen, will be broken down into disaccharides and then monosaccharides. Can you give me examples of carbohydrates? Sure. Starch, which is of plant origin, and glycogen from animal origin. The disaccharides include maltose, sucrose, and lactose. And then you break down the disaccharides into monosaccharides. So the maltose will give you glucose and glucose, sucrose will give you glucose and fructose, and lactose will give you glucose and galactose. Then you're going to break down the big proteins into polypeptides, oligopeptides, dipeptides, and amino acids, or monopeptides, so to speak. So dipeptide has 2 amino acids. Tripeptide has 3 amino acids. Oligopeptide has a few amino acids. Polypeptide has many amino acids. Then you break down the fat, which is big in the beginning, triglycerides, into smaller parts, such as glycerol and free fatty acids. It's not glycerol per se, but 2-monoacylglycerol. When it comes to digestion, your pancreas is the hero, because your pancreas can complete digestion from A to Z. Your pancreas has digestive enzymes to break down carbohydrates, to break down proteins, and to break down fat. Can you give me an example of an enzyme that breaks down carbohydrate? Sure. How about amylase? Oh yeah, that will do it. Next, can you give me an example of an enzyme that will break down proteins? A peptidase. Trypsin can also help. And to break down fat or lipid, I need a lipase. If it ends in ase, it's an enzyme. Metabolism came from the Greek meta, or metabole, which means to change. Oh, what type of metabolism? Well, I have 2 types. There is anabolic, to build up, and there is catabolic, to tear down. Let's build up. Okay, building up is an endergonic process. What do you mean? Endergonic means energy enters. Oh, so I need energy. Energy goes in. Energy goes in so that I can build up stuff, so that I can build up small glucose molecules into a big glycogen molecule. This is going uphill to build up, which requires energy. However, catabolism is the opposite. It's exergonic because energy will exit. It is flowing downhill because we are releasing energy. We are breaking down the glycogen into glucose, for example. This releases energy. Exergonic. Energy exits. When do you build up stuff? When I am fed. When do you break down stuff? When I am fasting. Who is the hero of the anabolic gland? It's insulin. And who is the hero of the catabolic gland? Glucagon and his friends. What do you mean by his friends? I mean epinephrine, cortisol, and sometimes thyroid hormone. Insulin alone is on the left, and every other hormone that you can imagine is on the right. So we say that insulin is a regulatory hormone, whereas the rest of the bunch are counter-regulatory hormones. Now let's talk about the most important topic in biochemistry, which is insulin land versus glucagon land, the feeding state versus the fasting state, the land of abundance versus the land of scarcity. This is the insulin world on the left, and this is the glucagon world on the right. Again, it's not just glucagon, it's glucagon and his friends. the epinephrine, the thyroxine, the cortisol, etc. Dear insulin, when do you work? I work in the feeding state because I am a builder. I am anabolic. I will build up proteins. I will build up glycogen. I will build up lipids. Tell me more. How do you build up proteins? I convert amino acids into big proteins. Hashtag proteogenesis, meaning protein synthesis. Proteo means protein. Genesis means synthesis, creation, formation. Next, glycogen anabolic. I take the glucose molecules and then build them up to glycogen. Hashtag glycogen synthesis or glycogenesis. That's if you want to build up glycogen. But if you want energy right now, I can burn that glucose for you via glycolysis to give you energy in the form of ATP or adenosine triphosphate. Speaking of fat, it's the same thing. I am fat anabolic because I will build up free fatty acids into big lipids, big triglycerides. Hashtag lipogenesis or lipid synthesis. Whereas glucagon and epinephrine and cortisol and thyroxine are destroyers. They work in the fasting state. When you're fasting, you're not eating by definition. So you need sources of energy. What should I do? You should break down what you stored for all these years. So the protein will be broken down into amino acids. Hashtag proteolysis or protein breakdown. Then you can take those amino acids and make glucose. Oh, you're making glucose from non-carbohydrate sources. Oh, you're making glucose from new sources? Gluconeogenesis. Genesis means formation. Neo means new. Oh, new, because this is non-carbohydrate source. Gluco, because I'm making glucose. Next, glucagon is glycogen catabolic. It will break down glycogen into glucose, which is the exact opposite of this. When you break down glycogen to glucose, this is called glycogenolysis or glycogen breakdown. Glucagon and his friends is also fat catabolic. It breaks down the big lipid triglycerides into free fatty acids and glycerol. Hashtag lipolysis or lipid breakdown. By the way, anytime you break down fat like this, ketone bodies are going to emerge, whether you like it or not. So you only see ketone bodies in the glucagon world. You never see ketone bodies in the insulin land, because insulin is the major anti-ketogenic hormone in the entire body. Never ever forget this. If there is insulin, there are no ketones. But if there is no insulin, there are ketones. And that's why some patients who have diabetic ketoacidosis, i.e. they lack insulin, guess what they do have? A bunch of ketone bodies. And these ketone bodies are what? Acetone, acetoacetic acid, and beta-hydroxybutyric acid. Have you noticed that these are acids for the most part? So ketones are ketoacids. And that's why if I have a bunch of these in my body, I have ketoacidosis. And since I have no insulin, they call me diabetic. So now I have diabetic ketoacidosis because I lack insulin. Therefore, I have a bunch of ketoacids in my blood. See, biochemistry makes so much sense once you understand what the French toast you're talking about. For the pros, please note that glycolysis belongs to the insulin world. But gluconeogenesis is part of the glucagon land. If you want to download these doozy colorful notes, go to medicosisperfectionalis.com. If you want me to personally tutor you, reach out to me on my website. This is about half of your biochemistry textbook in one slide. What is the central hub in biochemistry? This is acyl-CoA. It's the sun around which all the planets revolve. Because whether you eat proteins or lipids or carbohydrates, at the end of the day, they become acyl-CoA, the central hub. And then acyl-CoA, if you have oxygen, is going to delve into the TCA cycle, or the tricarboxylic acid cycle, also known as Krebs cycle or citric acid cycle. And then this will give you ATP. Moreover, you're going to capture the H in the NADH and FADH2. All of these hydrogens will be captured in the electron transport chain, which will give you even more energy. Both the TCA cycle and the electron transport chain occur in the mitochondrion. When you take glucose and burn it, it's called glycolysis, and this is part of the insulin world. Glucose gets burned into pyruvate, and then pyruvate becomes acyl-CoA by the famous enzyme pyruvate dehydrogenase. All of this started from big carbohydrates like glycogen, which is polysaccharide. Then you break it down into disaccharide and then into glucose and then glucose to pyruvate and then pyruvate to acetyl-CoA, and you enter the TCA cycle. How about proteins? They get broken down into polypeptides, oligopeptides, a bunch of amino acids, and boom, acetyl-CoA. How about lipid or fat? These triglycerides will be broken down into free fatty acids and glycerol. The glycerol can give you glucose because glycerol is gluconeogenic. The free fatty acids by beta oxidation will give us acetyl-CoA. Anytime you break down lipids, you're going to get ketone bodies. Ketone bodies can also give you acetyl-CoA, and acetyl-CoA can give you ketone bodies. When you are making ketone bodies, it's called a ketogenesis. When you are breaking down ketone bodies, it's called ketogenolysis. When you are making glycogen, it's called glycogen synthesis or glycogenesis. But when you are breaking down glycogen, it's called glycogenolysis. When you take some protein sources and some fatty sources and convert them to glucose, it's called gluconeogenesis. Some important facts. Carbohydrates, when you eat them, are going to give you 4 kilocalories per gram. Proteins, 4 kilocalories per gram. Fat, 9 kilocalories per gram. Alcohol, 7 kilocalories per gram. Notice that fat is 9, which is the highest number here. And that's why the favorite method for your body to store energy is in the form of fat. And that's why if you eat too much, you're gonna grow more obese. Oh, that's more fat. Why isn't it the case that when I eat so much, I become more muscular or more sweet? Why do I always become obese? Because fat is so dense from an energy standpoint. It gives you 9 kilocalories per gram. And that's why it makes more economic sense to store extra energy in the form of fat. That's how your body does it. Please take a moment to pause and review. Next, we'll talk about the amino acids. Start with this main carbon known as the alpha carbon. If you want to know the difference between alpha carbon, beta carbon, and gamma carbon, please refer to my organic chemistry playlist. Why do we call them amino acids? Because this is amino group and this is carboxylic acid group, amino acid. And this is the order by which they come out of the ribosome. Amino comes before the acid, that's why we say amino before the acid. And then there is a side chain or the R group, as you are familiar with from organic chemistry. And this will determine the properties of the amino acid. Some amino acids have H instead of an R. Some have methyl. Oh, just methyl? Yeah. Some have very long side chains. Some have branched side chains. And some have a ring structure. Please note that both the amino group and the carboxylic acid group are attached to the same alpha carbon. Amino is usually basic. Carboxylic acid is by definition an acid, and that's why we say that the amino acid is amphoteric, like an amphibian. It is half basic and half acidic. By the way, the amino could be written in many ways. You can write it NH2 or NH3+. Similarly, the carboxylic acid group could be written as COOH or COO negative. When should we write it as COOH versus COO negative was discussed in great detail in my video titled Titration of Amino Acids. Hint, it depends on the pH that you put the amino acid in and the pKa of the amino acid. Next, stereochemistry. Levo means left, dextro means right. There is also S versus R. Don't forget that in your body you have D sugars, but you have L amino acids. Sugars to the right, but amino acids to the left. What's the difference between this and this? Levo versus dextro is performed via plain polarized light rotation, but S versus R is done by a pencil and paper. And they were discussed in great detail in my organic chemistry playlist. Look at this. This is on the left, so we call this levo. But this one is on the right. We call it dextro. Keep in mind, humans have D sugars but L amino acids. So in your body right now, your sugars are D. For example, D glucose, not L glucose. But your amino acids in your body right now are L amino acids, not D amino acids. Do we have exceptions? Sure. All amino acids in your body are chiral with L, meaning to the left, meaning levo, except glycine, which is not even chiral, therefore it's neither L nor R. And cysteine is another crazy exception. Look, the amino acids in your body are L and S. Okay, but cysteine is L yet R. So when you use the plain polarized light, cysteine is going to rotate the light to the left, hence LEVO. But when you do it with pen and paper, you find it R, because it's clockwise and not counterclockwise. All the amino acids have the amino group and the carboxylic group attached to the same carbon. We call it the alpha carbon. However, keep in mind that we have something called GABA. What is GABA? Gamma aminobutyric acid. You know why we call it gamma? We call it gamma because the amino group is bonded to the gamma carbon, not the alpha carbon. How many amino acids are there? Lots and lots of amino acids. But hey, my professor said that there are only 20 amino acids. Your professor lied to you. Look, the 20 amino acids that you're talking about are the proteogenic amino acids. However, these are not the only amino acids in town, because we also have non-proteinogenic amino acids. What's the difference? Proteogenic amino acids get incorporated into proteins, hence proteogenic. They make proteins. But non-proteogenic amino acids are not incorporated into making proteins. Moreover, proteogenic amino acids are coded for by a genetic codon, a piece of the DNA code for alanine, for example, or valine, or glycine. But you do not have a code on your genetic codons to make ornithine. You do not have an order or a gene to make homocysteine or selenocysteine. But these are still called amino acids, and some of them are inside your body right now, such as homocysteine. Now, what are the famous 20 amino acids? These are alanine and arginine, aspartic acid, glutamic acid, aspargine, glutamine, phenylalanine, tyrosine, glycine, lysine, leucine, isoleucine, cysteine, valine, histidine, serine, proline, tryptophan, threonine, and methionine. Each one of these 20 proteogenic amino acids have a name, a 3-letter abbreviation, and a 1-letter abbreviation. For example, alanine is ALA, it's also just A. Lysine is LYS, it's also K. Isoleucine, ILE, it's also I. Histidine is HIS, also H. Take a moment to pause and review. Most professors, most exams want you to know their names and the 3-letter abbreviation. However, some professors happen to hate their mothers. They want you to memorize everything, including the 1-letter abbreviation. These people are sick. Next, some amino acids can give you glucose. We call these glucogenic. Some amino acids can give you ketone bodies. We call these ketogenic. Some amino acids can give you both glucose or ketone bodies. We call them both glucogenic and ketogenic. What are the amino acids that are purely glucogenic? These include alanine, cysteine, methionine, arginine, aspartic acid, glutamic acid, aspargine, glutamine, proline, valine, glycine, histidine. Which ones are purely ketogenic? Lysine and leucine, leucine and lysine, lysine and leucine. Which ones can give you both glucose or ketone bodies? Isoleucine, phenylalanine, tyrosine, tryptophan, threonine, and serine. Notice that almost all of the amino acids can give you glucose. And these are all the amino acids except lysine and leucine. Another classification of amino acids, we have essential amino acids, semi-essential, also known as conditionally essential amino acids, and non-essential amino acids. The essential amino acids, what does that even mean? Essential means that your body cannot make them from scratch. Therefore, it is essential that you eat them and consume them in your diet. And these include valine, leucine, isoleucine, phenylalanine, tryptophan, methionine, threonine, histidine, and lysine. On the other hand, we have some non-essential amino acids. It means that your body can make them from scratch, therefore you do not have to eat them in your diet. These non-essential amino acids include alanine, aspartic acid, glutamic acid, aspargine, and serine. Then we have the semi-essential or conditionally essential. If you are a normal person, you do not need to eat them in the diet. They are not essential because your body can make them. But if you suffer from a certain metabolic disease, such as the famous inborn errors of metabolism, then one or more of these amino acids become essential because of the disease that you have. For example, there is a disease known as phenylketonuria. In this phenylketonuria disease, tyrosine, which was normally non-essential, becomes essential only in patients with phenylketonuria. So, if most people did not eat tyrosine in the diet, they will be okay. But if a patient with phenylketonuria forgot to consume tyrosine in the diet, this patient will suffer big time, because for that phenylketonuria patient, tyrosine is essential. Others include arginine, cysteine, glycine, glutamine, and proline. To learn more about phenylketonuria and the other inborn errors of metabolism, please refer to my biochemistry playlist. Now let's classify these amino acids in another way. You can classify them into 5 groups as you see here. First group, non-polar non-aromatic side chains. Second group, aromatic side chains. And these will have a benzene ring, hence aromatic. And these are beloved by your chef, Gordon Ramsay. Oh, F me, it's aromatic. In my 30 plus years in the culinary industry, I've never seen so much arrogance. Hey, big boy, listen. Get your head out of your sphincter. Pathetic. Number 3, polar non-aromatic side chain. Number 4, negatively charged or acidic side chain. And last, the amino acids that are positively charged or with basic side chain. Groups 3 and 4 and 5 can form hydrogen bonds with water in aqueous solutions. Here are the 5 groups again. Group number 1, non-polar non-aromatic side chain. These include alanine, glycine, methionine, proline, and the branched chain amino acids, including valine, leucine, and isoleucine. Then we have the aromatic side chain. They have a benzene ring. These are the phenylalanine. If you add hydroxyl to it, it becomes tyrosine. And you also have tryptophan. Phenylalanine, tyrosine, tryptophan. Then you have polar non-aromatic side chain. Some of them are hydroxyl-containing amino acids, the serine and the threonine. Some of them are amide side chain-containing amino acids, the aspargine and glutamine. Some of them contain sulfur, such as the famous cysteine. Negatively charged or acidic side chain, well, it's called acidic for a reason. I can say aspartic acid or glutamic acid. And they have an acid group. Remember, the carboxylic acid is COOH. If it's deprotonated, it becomes COO-. This is minus, hence negatively charged. On the other hand, we have positively charged or basic side chains. These include the arginine, the lysine, and the histidine. Now let's start by talking about the first group, which is the non-polar, non-aromatic side chain. These include alanine and glycine, the branched chain, valine, leucine, and isoleucine, methionine, as well as proline. Please keep in mind that methionine and cysteine are sulfur-containing amino acids. You should never ever forget this. Methionine and cysteine are sulfur-containing, whereas serine and threonine are hydroxyl-containing. I should be able to wake you up from a coma, and you should still remember that methionine and cysteine contain sulfur, whereas serine and threonine contain hydroxyl. Now get some sheets of paper and a pen, because I'm about to take you to class. We will draw every one of the 20 amino acids, and we will learn 3 facts about each amino acid. Starting with the amino acid glycine. How do you draw glycine? Just like any amino acid with the alpha carbon in the middle. Then you have amino on one side and acid or carboxylic acid on the other side. You have H here and you have the R here, except glycine is different. Glycine has H instead of an R. And that's why, according to legend, glycine is the smallest amino acid. Look at this cutie patootie. Why is it the smallest amino acid? Because it has H instead of a long R. It's the simplest, smallest amino acid. What are the 3 facts that you need to know about glycine? Fact number 1, it's the smallest, simplest amino acid. Fact number 2, it is achiral, because this carbon is bonded to hydrogen and hydrogen, making it not chiral. Because by definition, a chiral carbon needs to be bonded to 4 different species. But these are not 4 different species. That's why the C is not chiral. Therefore, glycine is neither levo nor dextro. It's neither L nor D. Fact number 3 is that glycine takes part in the formation of collagen, which is a famous member of the extracellular matrix of your body. We find collagen in bones, cartilages, tendons, the sclera of the eye, the basement membrane, etc. Non-polar, non-aromatic side chain. Next, alanine. How do we draw alanine? Same, with an alpha carbon in the middle. This carbon is alpha. It is not a beta soy boy. Nuh-uh, it is alpha. And you have amino on one side, carboxylic acid on the other side. You have H here. And instead of an H like glycine, alanine is more complex. It has an actual R, which is just methyl. Alanine, another nonpolar, non-aromatic side chain amino acid. What are the 3 facts that you need to know about alanine? Fact number 1, you can convert pyruvate into alanine and alanine into pyruvate. The famous enzyme that does this is called alanine transaminase or alanine aminotransferase, or simply ALT, which requires vitamin B6, pyridoxine, as a cofactor. Never ever forget this. ALT is very important in your liver. It's one of the liver enzymes or liver transaminases. Fact number 2, that alanine is part of something called the Cahill cycle. This is the hill that alanine wants to die on. So now you know the 3 facts of alanine. Fact number 1, you can convert it to pyruvate and you can convert pyruvate to it. Fact number 2, this is the Cahill cycle. Fact number 3, I need ALT and B6 for this. Next, we have methionine, still nonpolar, non-aromatic. How do I draw methionine? Alpha carbon here. You have amino on one side, carboxylic acid on the other side. Give me that hydrogen here. And then what? CH2. Am I done? Nuh-uh. CH2. Am I done? No. Methionine is a sulfur-containing amino acid. Don't forget your sulfur. And end with a methyl group. This is methionine. Methionine. It is sulfur-containing. What are the 3 facts that I need to know about methionine? Fact number 1, it contains sulfur. Fact number 2, remember the start codon on your beautiful mRNA? Remember AUG? AUG codes for methionine, the first amino acid to be put in the chain. Fact number 3, that methionine can give you Uncle Sam. Uncle Sam is the ultimate methyl group donor. And when Uncle Sam loses its methyl group, it becomes Uncle Sa. S-adenosylmethionine versus S-adenosyl-homocysteine. Here is glycine, the simplest amino acid. Here is alanine. Instead of the H, you have methyl. Here is methionine, which has sulfur, and it has methyl, methionine. Then we'll talk about branched chain amino acids. We are still talking about nonpolar, non-aromatic side chains, but we're going to add a branch. So let's draw valine together. How do you draw valine? Easy. Here is your carbon. Here is the amino, and here is the acid. And then what? You have an H here. Okay, but look at the branch that's going to happen. Oh, look at this branch. This is a branched chain amino acid. And you have a methyl here and a methyl there. This is valine. What are the 3 facts that I need to know about valine? Fact number 1, it is a branched chain amino acid. Fact number 2, it is the smallest branched chain amino acid. And fact number 3, there is a disease known as maple syrup urine disease, where the patient has deficiency in the branched chain alpha keto acid dehydrogenase. These patients cannot metabolize valine, leucine, or isoleucine. So valine, isoleucine, and leucine will end up in the urine, giving the urine the classic maple syrup odor. This disease is known as maple syrup urine disease. Next amino acid is leucine. How do we draw this? It is very similar to valine, but a little more complex. So here is the amino, and here's the carboxylic acid. This is the hydrogen, and this is the R. You have CH2. And then you're gonna do CH, then you branch. Oh, so what is the difference between valine and leucine? The difference is just here, that valine had CH and then you branch. But leucine has CH2, then CH, then you branch. So leucine is more complex. What are the 3 facts that you need to know about leucine? Fact number 1, it is a branched chain amino acid. Fact number 2, it is muscle essential. Fact number 3, because it's branched, it is also implicated in maple syrup urine disease. Then we have isoleucine. Iso means what? Same or similar to leucine. Oh, so it's going to be similar to leucine, which means it's branched as well. So here is amino on one side, here is the carboxylic acid on one side, and here is H, then the R. Look at this. The R is branched in a different way. You have the H here, and you have the CH3 here, and then you continue, CH2, and CH3. The carbon that's attached to the amino and the carboxylic is the alpha carbon, and it's going to be followed by the beta carbon. Is this carbon chiral? Well, here is something, 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 and all of these are different. The species are different. So this is bonded to 4 different species. So yes, this is a chiral center. How about this? Well, I am bonded to H on one side, CH3 on one side, CH2 on the third side, and C, which is bonded to all of these on the other. So yes, I am chiral, meaning that isoleucine has 2 chiral centers. So what are the 3 facts that we need to know about isoleucine? Fact number 1, it is branched. Fact number 2, it has 2 chiral centers. Fact number 3, because it's branched, it's implicated in maple syrup urine disease. These are the branched chain amino acids. Valine is the simplest one, then leucine, then isoleucine. Isoleucine has 2 chiral centers. To learn more about chirality, please refer to my organic chemistry playlist. Next, proline. Proline is cyclic. It has a ring. So let's draw. Well, this is the alpha carbon, and here's the H. Here is COOH on one side, and NH2 here. And then what? Then this carbon is attached to CH2, which is attached to CH2, and which is attached to CH2, and boom, before you know it, we have this ring. It is a 5-membered ring, as you see. And this 5-membered ring has nitrogen, as you see. Therefore, we call proline, it's not just an amino acid, but an amino acid as well. What are the 3 facts that you need to know about proline? Fact number 1, it is cyclic. It has a 5-membered ring. Fact number 2, proline with a P is a pervert with a P. Why? Because this ring introduces kinks in the peptide chain, and that's why it's a pervert. And because it's a pervert, it's not only an amino acid, but also an amino acid. Even the word amino was perverted. Fact number 3, you need proline to make the famous collagen, which is found in bones, cartilages, tendons, blood vessel walls, sclera, and more. Here is proline again. Please pause and review. This concludes the first group of amino acids, which is the nonpolar, non-aromatic side chain. The next group is aromatic side chain, meaning that they have a benzene ring. These include tryptophan and phenylalanine, which will make tyrosine. Get you a piece of paper and doodle with medicosis. How do I draw tryptophan? Easy. Here is your alpha carbon, and you have amino, you have acid. Good. Don't forget the hydrogen, don't forget the R. This R has a ring because we're talking aromatic here. So look at what's happening here. Okay. Look at what's happening here. Okay. Look at what's happening here. What is going on? Well, you gotta add a benzene ring just like this. This is just beautiful. Amazing. How many rings does tryptophan have? The answer is 2. It is a double ring. What are the 3 facts that I need to know about tryptophan? Number 1, it is the only amino acid that has a double ring. Fact number 2, you need tryptophan to make niacin. Niacin is vitamin B3, and this niacin, or nicotinic acid, will give us the NAD+, and if it gets reduced, it becomes NADH. And of course, you're gonna capture that H in the electron transport chain to make some energy. Fact number 3 about tryptophan. You need tryptophan to make serotonin, and you need serotonin to make melatonin. Why do we call serotonin 5-HT? Because it's 5-hydroxytryptamine. Tryptamine? Yeah, because it came from tryptophan. And tryptophan is an amino acid. Tryptamine. Next, phenylalanine, also aromatic, also has a ring. This is my alpha carbon. Here is amino on one side and carboxylic acid on the other side. Don't forget the H, don't forget the R. This R is CH2, and boom, you have your benzene ring, which is a 6-membered ring. And don't forget the double bonds. If you do not want to draw the double bonds, you can draw a circle instead. Even better. What are the 3 facts that you need to know about phenylalanine? Fact number 1, it's an aromatic amino acid with a benzene ring. Fact number 2, it is relatively non-polar. Fact number 3, phenylalanine will make you tyrosine if you just add a hydroxyl group to it. So phenylalanine plus hydroxyl group equals tyrosine. And that's why tyrosine is a semi-essential amino acid. If you're healthy, you do not have to eat tyrosine because, don't worry, your phenylalanine will make you the tyrosine that you want. But if you have phenylketonuria, you cannot make tyrosine from phenylalanine. Therefore, tyrosine becomes essential for you, which means you have to eat it in the diet. See what happens when you get your head out of your sphincter? You tend to understand biochemistry better. Next is tyrosine. Are you aromatic? Of course I am aromatic. Where did you come from? I came from tryptophan, which is the previous amino acid. If you just add a hydroxyl group to tryptophan, it will become tyrosine. So let's draw tryptophan, which looked like this, the famous benzene ring, which is amazing like this, and add to this OH. Boom, you have tyrosine. What's the name of the enzyme that added the hydroxyl group to the phenylalanine to make tyrosine? Well, guess what? Hydroxylase. No duh. And this phenylalanine hydroxylase enzyme is deficient in patients suffering from a disease known as phenylketonuria, which was discussed in detail in a separate video in this biochemistry playlist. What are the 3 facts that you need to know about tyrosine? Fact number 1, it's aromatic with a benzene ring. Fact number 2, it is relatively polar. Why? I added OH, making it relatively polar when you compare it to phenylalanine, which was relatively non-polar. Fact number 3, tyrosine will give us DOPA and thyroid hormone. And DOPA can give us dopamine. And dopamine can give us norepinephrine. And norepinephrine can give us epinephrine. Norepinephrine is also known as noradrenaline, and epinephrine is the same as adrenaline. So phenylalanine will give you tyrosine. Tyrosine will give you DOPA. DOPA will give you dopamine. Dopamine will give you norepinephrine. Norepinephrine will give you epinephrine. So it goes like this. Phenylalanine, tyrosine. DOPA, dopamine. Norepinephrine, epinephrine. Phenylalanine, tyrosine. DOPA, dopamine. Noradrenaline, adrenaline. All of this happens in the adrenal medulla. The last 3, dopamine, norepinephrine, and epinephrine, are famously called the catecholamines. Amines, yeah, because we're talking amino acids here. These are the aromatic side chains which have benzene rings. You have tryptophan, you have phenylalanine, and if you add hydroxyl to it, it becometh tyrosine. What's the name of this enzyme? Phenylalanine hydroxylase. And this is the same song that we just did. Phenylalanine, tyrosine, DOPA, dopamine, norepinephrine, epinephrine. Or phenylalanine, tyrosine, DOPA, dopamine, noradrenaline, adrenaline. All of this happens in the adrenal medulla. Dopamine, norepi, and epi are collectively known as catecholamines. Tyrosine can give you DOPA. It can also give you thyroid hormone in your thyroid gland. Phenylalanine is converted to tyrosine through a famous enzyme known as phenylalanine hydroxylase, which is missing in patients with phenylketonuria. Then from tyrosine to DOPA, there is a bunch of enzymes and there is a cycle. Enzyme deficiencies will give me tyrosinemia. DOPA can make melanin, and if I have a problem or deficiency in this enzyme, tyrosinase, I will not be able to make melanin and I will suffer from a disease known as albinism. An albino is a person with a very, 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 very light and pale skin because this patient cannot make melanin because this patient is lacking tyrosine. This concludes the second group of amino acids. On to the third group, polar non-aromatic side chain. These include serine and threonine. Both of them have hydroxyl group. We have cysteine, which has sulfur. What else had sulfur today? If you said methionine, you're absolutely correct. Then we have asparagine and glutamine, and they have amide. Notice that amide, I had a circle that is blue. You know why? Because amide has oxygen in it, and this is the O that I drew inside the D. Let's draw serine. Remember that this serine has hydroxyl. Yes, it does have hydroxyl. But does it have a benzene ring? No, these are non-aromatic amino acids. So I do not have a benzene ring, but I do have a hydroxyl group. So let's draw serine. Where should we start? We should always start with the alpha carbon. Give me an amino here and a carboxylic acid here. Don't forget your H and the R, where here's a carbon. I will not forget the OH because serine is hydroxyl containing. And then on the other side, H and H. Is this a chiral carbon? Well, since it's bonded to 4 different species, yes, indeed, it is chiral. However, look at this carbon. Is it chiral? Well, here is H, here is H. These are not different species. So the answer is no. This second carbon is not chiral, which makes it liable for all the damages that happen next. Next is threonine, another hydroxyl-containing amino acid. So here is the alpha carbon, here is amino on one side and carboxylic acid on the other side. Don't forget the H, and look at this. There is COH here and H there. But instead of an H as in serine, here you have a CH3. What are the 3 facts that you need to know about threonine? Number 1, it's hydroxyl containing, as you see. Number 2, it has a methyl group instead of an H. And the way I remember this is that I think of threonine with a TH as methyl group with TH. And number 3, unlike serine, threonine actually has 2 chiral centers. Serine versus threonine, both are hydroxyl containing. The difference is that threonine has a methyl group. Next we have cysteine. This one has sulfur, so I gotta find a way to put the sulfur. This is amino, this is acid, this is the H, and then CH2. Now you add the sulfur, SH. What are the 3 facts that you need to know about cysteine? Number 1, it is polar and non-aromatic. Number 2, it has sulfur, and because it has sulfur, we can use it to make disulfide bonds or disulfide bridges. And we need this in the primary and tertiary protein structure. To learn more about the difference among primary, secondary, tertiary, and quaternary protein structures, there is a video on this topic which you can find in this biochemistry playlist. Here is cysteine and it has sulfur. Next is asparagine. Okay, asparagine is one of those that has amide. What's an amide group? A group that has nitrogen and oxygen. So let's just start by amino on one side and acid on the other. Don't forget the H, and then you have CH2 here. And where is the amide? The amide is C, double bonded to oxygen, and the same carbon is bonded to nitrogen. This is amide. The 3 facts that you need to know about asparagine include the fact that it's polar, the fact that it has non-aromatic side chain, and the fact that we use it for strong hydrogen bonding in the secondary protein structure. Next is glutamine, also have an amide group, so it's going to be very similar. Here's the carbon, here is amino on one side and the acid on the other side. Here is the H, and then CH2, repeat it again, CH2, and then give me that amide group, C double bonded to O and bonded to nitrogen. So simply, glutamine is slightly longer compared to asparagine. What are the 3 facts that you need to know about glutamine? Number 1, it is polar. Number 2, has non-aromatic side chain, and number 3, it is important as a transport of nitrogen throughout the body. And this is going to be super important when you learn about transamination, deamination, and the urea cycle later in this playlist. Here is asparagine, which has an amide group, and this is glutamine, another amide group. This concludes group number 3. Next, group number 4, negatively charged or acidic side chain. Aspartic acid has an acid, COOH. This is an acid. What if we remove the H? What if that proton left the chat? The remainder will be COO negative. We call them acidic because this is an acid. We call them negatively charged because COO negative. Aspartate came from aspartic acid after it lost a proton. But when glutamic acid loses a proton, it becomes a glutamate. And if you are a chemistry buff, you would recall that if something ends in 8, odds are it is negative. Does anyone remember bicarbonate? Negative, because it ends in 8. Note that aspartate and glutamate contain CO negative, but asparagine and glutamine contain amide side chain. How do we draw aspartate, which is negative? This is how we draw it. This is the carbon, amino on one side, and the carboxylic acid on the other. And here's the H, CH2 is here. And then where's the COOH? It is COO, but I lost the H. So instead of COOH, it is COO negative. it is a negatively charged side chain. The 3 facts that you need to know about aspartate. Number 1, it came from aspartic acid after it lost a proton. Number 2, it has a negatively charged side chain. Number 3, it helps eliminate nitrogen in the urea cycle. Later, when we talk about the urea cycle, you will know that urea has 2 nitrogen groups. One of those nitrogen groups actually came from aspartate. Never ever forget this. Next is glutamate, similar to aspartate but longer. So here is the C and here is amino on one side, carboxylic acid on the other, here's the H. The side chain is longer, so CH2, another CH2, and then you have the carbon bonded to oxygen and O negative. This was COOH and we called it glutamic acid. After losing a proton, it became COO minus. The 3 facts that you need to know about glutamate. Number 1, it is derived from glutamic acid after a proton was lost. Number 2, it has a negatively charged side chain. Number 3, glutamate is actually an excitatory neurotransmitter in your nervous system. Remember that GABA is inhibitory, but glutamate is excitatory. Here is aspartate and the longer glutamate. Pause and review. This concludes group number 4. Last, group number 5, positively charged or basic side chains. We have arginine, which has 3 nitrogens. We have lysine, and we have histidine with the famous imidazole ring. Mnemonic to help you remember these 3 amino acids is the following. Her leggings are, so basic. Basic because these are basic side chains. HLA, histidine, lysine, arginine. For the pros, you know the histones? Oh, DNA. Yup. We wrap your DNA around histones to make chromosomes. These histones are nucleoproteins, nucleic acid and proteins. These histones are rich in arginine and they are rich in histidine. Histidine, histones. Arginine has 3 nitrogens. Look at all the N's in the word. Here's the alpha carbon, which is bonded to amino on one side and carboxylic acid on the other. Don't forget the H, don't forget the R. What is the R? Very long. CH2. Repeat it. CH2. Again, CH2. Then a nitrogen bonded to hydrogen here and to a carbon here. Carbon is double bonded to nitrogen and single bonded to nitrogen. Here is a nitrogen, here is the second nitrogen, and the third nitrogen. What are the 3 facts that you need to know about arginine? Number 1, it's positively charged. It has a basic side chain. Number 2, arginine can give you something called nitric oxide, which is a potent vasodilator. It dilates your blood vessels. Number 3 is the fact that arginine has 3 nitrogens. Next, we have lysine, similar to arginine but less drama. This is the alpha carbon, and this is my amino, and this is my acid. Don't forget the hydrogen, don't forget the R. CH2, CH2, repeat it again, CH2, repeat it again, CH2, and finally the nitrogen. Is this basic? Of course, this is basic. Is this positively charged? Yup. What are the 3 facts that you need to know about lysine? Number 1, it is basic. It is positively charged side chain, as you see. Number 2, it has a terminal primary NH3 group. Number 3, later in fatty acid metabolism, you'll learn about the carnitine shuttle. This lysine is going to be converted to carnitine to help fatty acid transport in the carnitine shuttle. To learn more about fatty acid metabolism, please refer to my videos in this biochemistry playlist. The last amino acid for today is histidine. Histidine has nitrogen and it also has a ring. So, this is the carbon. It's called the alpha carbon. This is NH2 and this is the carboxylic acid. And don't forget the H, don't forget the R. Here is CH2, and then we're going to start building the ring. Here is carbon, double bonded to another carbon. Single bonded to nitrogen, which has hydrogen here. Double bonded to carbon, which has hydrogen here. And single bonded to nitrogen, and then you continue the ring. And this is called imidazole ring or imidazole. What are the facts that you need to know about histidine? Number 1, it is positively charged basic side chain. How come? This is the positive end here. And of course, this end could be positive if you protonated, but this depends on the pH, and I've talked about this topic in my video called titration of amino acids. Fact number 2 about histidine. it has an imidazole ring. And of course, amide has nitrogen, as you see. Fact number 3. Histidine is a physiological buffer in your body. Because you know what the pKa of this group is? It's about 6. And what's the pH of your body? It's about 7.4. Since the pK is similar to the pH, this makes histidine a very good buffer. And don't forget that you need histidine in histones. Here are the positively charged basic side chain amino acids. You have arginine, lysine, and histidine. The mnemonic is her leggings are so basic. And here are the 5 groups of amino acids again. Please pause and review. Amino acid derivatives: phenylalanine, tyrosine, doped, dopamine, norepinephrine. Glycine will give me protoporphyrin, which gives me heme, which is part of the hemoglobin, which carries oxygen and carbon dioxide in your blood. Tryptophan gives me serotonin, which gives me melatonin. It also gives me niacin, and niacin gives me NAD+. If you reduce it, it becomes NADH. If you capture this H in the electron transport chain, you're going to end up with energy. How many ATPs? About 2.5, approximated to 3 ATP molecules per 1 molecule of NADH. Histidine gives me histamine by decarboxylation. Decarboxylation, meaning removal of carbon dioxide. Also, glutamate will give me GABA when carbon dioxide leaves the chat. The moment you say decarboxylation, vitamin B6 is going to help you as a cofactor. In order to make collagen in your bones, in your tendons, in your cartilage, in your blood vessels, in your basement membrane, in your hair, in your sclera, etc., you need 3 amino acids. Don't forget them: glycine, proline, and lysine. Quick note on protein metabolism. Again, metabolism has 2 parts: anabolism and catabolism. Don't forget that anabolism is insulin land, but catabolism is glucagon land. You take those amino acids and then bind them together. Oh, dipeptides, then tripeptides, then oligopeptides, many polypeptides, bigger proteins. Amino acids, just 1 amino acid. Get 2 together, bind them, you have dipeptide. Get 3 together, tripeptide. Get between 4 and 20, oligopeptides. More than 20, polypeptides. What are you trying to achieve? You should begin with the end in mind. But what end are we talking about? Are we talking about the N terminus or the C terminus? Haha, very funny. That's the funniest that you can get with biochemistry. Of course, the N terminus is this and the C terminus is this, amino acid. What I was trying to say is, what are you trying to achieve? Are you trying to build up? This is anabolic. Or to break down? Catabolic. If you want to build up, let's lump some amino acids together, combine them together, condense them together to make bigger proteins. How do you condense? By removing water. It's a dehydration reaction. So making peptide bonds is a dehydration or condensation reaction. But breaking down proteins into amino acids requires the opposite. Instead of removing water, you need to add water, and this is called hydrolysis. So, forming peptide bonds is a condensation dehydration reaction. But breaking down peptide bonds is a hydrolytic reaction performed by hydrolytic enzymes, such as your digestive enzyme. Digestion is catabolism. So here's 1 amino acid, here's another amino acid. Let's lump them together to form a peptide bond. This used to be COOH. The OH is going to leave the chat. This used to be NH2. One of the H's will leave the chat. H plus OH, boom, water. Water is leaving. Hashtag dehydration, hashtag condensation. Condense them together. What's the name of this bond? Peptide bond. How does it look like? It's an amide group. It's a carbon double bonded to oxygen, and the same carbon will be single bonded to that nitrogen, and that same nitrogen is bonded to hydrogen. This is amide for you. Amide has oxygen. Let's make a peptide bond. Here's 1 amino acid. Here's the second amino acid. Remove water. Hashtag condensation. Hashtag dehydration. Before you know it, you have a dipeptide. You have a peptide bond. The peptide bond has an amide group. Can you call this condensation? Sure. Can you call it dehydration? Sure. Can you call it acyl substitution? Also sure. To learn more, refer to my organic chemistry playlist. Here's 1 amino acid. Here's another amino acid. Let's make a peptide bond. Make a peptide bond. Before you know it, I have a dipeptide. What is this? Carbon double bonded to oxygen. And the same carbon is bonded to nitrogen, which is bonded to hydrogen. This is amide group, which is the essence of the peptide bond. Remember, if you want to make a peptide bond, if you want to build up proteins, this is anabolic, insulin world. But if you want to break down the peptide bond, this is catabolic, glucagon world. Peptide bond formation is condensation or dehydration, but peptide bond breakdown is hydrolysis. Remove water, add water. Now let's break down proteins into amino acids. You do this every day you eat proteins. When you eat proteins, your body's going to add water to them during digestion in your intestines. The proteins become dipeptides. When you add water, it's called hydrolysis. The enzymes are hydrolases, such as the peptidases, such as the trypsin, the chymotrypsin, carboxypeptidases, etc. Add another water before you know it, I have amino acids. This is hydrolysis, which is breakdown. The trypsin and the chymotrypsin are hydrolytic enzymes or hydrolases. Trypsin breaks the carboxylic end or the C-terminus of arginine and lysine, but chymotrypsin breaks the carboxyl end of phenylalanine, tyrosine, and tryptophan. The way I remember which is that I think of chymo as chemical, and chemicals can smell like perfumes. And these are what? Aromatic amino acids. They smell. How do you break a peptide bond? You add water. Add OH here is going to attack on this side, and add an H here to attack on that side. This is basically the opposite of peptide bond formation. Instead of condensation, we're performing hydrolysis today. You need to hydrogen the nitrogen and hydroxyl the carboxyl. Hydrogen the nitrogen and hydroxyl the carboxyl. Hydrogen the nitrogen, hydroxyl the carboxyl. Before you know it, I'm breaking the peptide bond, #hydrolysis, and I have 2 separate amino acids now. To learn more about all of these different digestive enzymes, please refer to my physiology playlist, especially the gastrointestinal physiology series. During periods of prolonged starvation, your body has to do something. Break down sugar. We did this a long time ago. Break down fat. Done. What else should we do? Break down proteins. Your body does not want to do this, but your body was forced to do this with its back against the wall because you're starving, and I better break down my muscles than die. So I'm going to break down the proteins, including the proteins in the muscle. So, catabolism of proteins will happen. Is this insulin land or glucagon land? Glucagon land, of course. When you break down proteins, they become amino acids. By transamination and deamination, amination, NH2 will leave, okay? And then what? This amino will enter the urea cycle, and the ammonia will be converted to urea, and urea will leave the body through the kidney. As for the remaining carbon skeleton, we can use this to release energy. If the amino acid was one of those glucogenic amino acids, voila, we can make glucose. If it was a ketogenic amino acid, we can make ketone bodies. And this was the story of biochemistry in a nutshell. I hope you learned something today. If you value what I do, help me make more videos by supporting the channel. Go to buymeacoffee.com/medicosis. There are more than 600 premium videos available on this channel when you click the join button and choose the highest tier. Please subscribe, hit the bell, smash like, support my channel on Patreon, PayPal, or Venmo. Go to my website to download my courses, notes, and cases, and if you would like me to tutor you. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine, chemistry, math, and physics make perfect sense.